Hello everyone. I'm Justin Priest. I'm a research assistant in Pongich Jaiswal's lab at Oregon State University, and I have the good fortune to be working on the Gramene project. Today I'll be giving a pretty brief overview of the existing system so that I can move quickly to some of the exciting work we've been doing in the last year. And in the new version shortly, we're going to be showing gene expression overlays, doing visual species comparison, and other analysis tools. For a review of the basic premise of the Reactome data model, since we are sharing code and have been the beneficiaries of software from the human Reactome project, the representational data model in Reactome is to put the reaction at the center of the activity, and crucially, Reactome spent a lot of time making sure that you could visually represent both regulation and catalytic activity at that level, and have good, precise, diagrammatic icons for binding and activation, translocation, and degradation. So you can show a lot of detail in your pathway. From the Gramene site, you can get to the pathways through the icon on the right or the link on the left. We do continue to host, I just want to point out, the site platform pathways. We're in the process of moving those to the iPlant environment. In fact, it's already there and I think undergoing active testing. Will be publicly available soon. But we're spending a lot of our time and focus on Plant Reactome. So if you click through on the icon on the right, to give you a very brief tour, this would be the homepage of the current live site. It's also accessible at plantreactome.gramene.org. And from here you can access a pathway browser that I'll show you in a moment. There are boxes for doing quick keyword searches for elements in the pathways. We let you know what we're up to most recently here, and this provides navigation bar access to downloads. Both the full database is downloadable, as well as summary tab files of our gene products and their relationships to pathways by species. And our projection statistics are always available under our documentation as well. The pathway browser itself. I'll just highlight a few things. On the right, you've got a zoomable, draggable interface to explore the pathways visually. On that, you'll find many familiar elements, your reaction locations, the catalysts that are responsible for driving those reactions, the inputs and outputs, as well as various small molecules that are involved. At the bottom, as you highlight any particular element on the pathway, you can see the details about that element at the bottom, including link outs to things like Uniprot, Ensemble via Gramene, and in some other cases, some specialized species databases such as Maze GDB. On the left-hand side, you've got navigation to the pathway hierarchy grouped by pathway category for each species. And at the top, you've got a species switcher here. If I were to drop that down, you'd see 33 different species listed off to our projection data. More about that in a bit. There are more advanced search capabilities as well. You could drill in, say for example, and look at just all of the pathways and limit that to those that contain ethylene and the name. And say you want to limit your species and just look at Oryza rufipogon, for example. You could do that, and it would return the results to you that are existing in our database. And then you could click through from those results and it would take you to the Rufi ethylene biosynthesis pathway if that was your selection, and you could get more detail from there. We do have other search features and downloads. You could do a small molecule search, which involves the KEBI database, and it will return molecular diagrams and then also cross-links back into our Plant Reactome database showing you where those molecules appear in our curated pathways. You have more granular download options while you are on a particular pathway. You can go to the detail pane at the bottom and select just for the proteins, gene products, or chemical compounds in that pathway and download that specific information, including selecting which columns or fields you would like to see and what format you would like to get that information in. We also have, for programmatic access, similar to the sorts of things, SBML downloads available by pathway. You could get this information if you were interested in writing software against what we have available on our live site. moving ahead to some of the things we've been doing in the last year, orthology projection. We have projected from Oryza sativa to 33 other species using Reactome's orthology inference software. This is the same software that the Human Reactome project has used. We downloaded Ensemble Gramene's compara data for 27 species, as I mentioned earlier, monocots and dicots, as well as some published leaf tissue transcriptomes. I believe they come from the IOMP project, Rod Wing's group at Arizona, and also Oryza casalith, which was published in a paper that I think I have a citation on in a moment. And we prepare those mapping files using some code we wrote in-house. It's always fun to mash together names of other people's programs, so we call this one InComparanoid. We'll add as many syllables as we can get away with. With that Python code, you'll see in many cases we end up with a one-to-many relationship. On the left-hand side, you've got uniprot IDs representing the Oryza sativa gene products. And then in this particular case, although it's very small, what we're mapping to is orthologs in maize using the compara data that we have downloaded. Running the orthology inference software that comes with the Reactome platform leads to an entire projected set of pathway data from our curated Oryza sativa data. We keep track of the statistics associated with that projection effort and publish this minus some of the color coding, but the data is there on the live website. I've color-coded it here for you so you can get a sense of the areas into which we're projecting and what the results are. On the left-hand side, you see the list of species. We have here the dicots at the top, the monocots follow, and including all of the Oryza taxa that I was mentioning. In each column, they're individually heat mapped, if you will, with blue being representing a high projection saturation. We had a good projection return rate after we set our thresholds for reciprocal identity amongst the compara data. And the red represents a lower return rate with yellow marking the intermediate. And on the left, I've also highlighted those in green were generated by imparanoid clustering instead of compara. There are some interesting results because of this. I want to make it very clear, we're not making any phylogenetic claims here at all. This heat map has nothing to do with phylogeny. It simply represents the projection results we are getting using the Reactome inference process. That having been said, when you get a low result like this for casalith, it probably just indicates low annotation more than anything else. And that as the quality of the annotation on that particular genome improves, or transcriptome, as the case may be, then we'll get better results in there. Another interesting thing is that amongst the imparanoid clusters, those numbers tend to come out marginally lower than the compara clusters within the Oryzas. And it would be interesting to do some control statistical studies to see how much of that is annotation-based, how much of that is algorithmically driven due to the differences between imparanoid clustering and compara. We've played around with that a bit, but we haven't done any rigorous control studies on that comparison yet. What's the purpose of projection? Well, it's obviously making interspecies comparison, and that's one of the purposes. And so you can right now on the live Reactome platform do that by moving amongst various pathways across species, say starting with Japonica, and then looking at the same pathway as projected into Indica, Rufi, Nivara, even something a little more distant like Arabidopsis. Right now, in this particular pathway's case, what that does is confirm that we have good compara data representing the orthology across these species. We've got a hit at all of the points of catalysis, which will fill out the rest of the pathway and confirm that we have good projection data. In another case, say with ethylene biosynthesis and signaling, if you do a similar comparative view and move from Japonica through Indica, you see that they're the same visually. But then if you jump to something like Australiensis, there's a portion missing. Just back up and move to it again. You'll see it on the upper right-hand side. That's because that particular catalyst or gene product was not represented in the orthology data, and therefore it's missing from the Australiensis diagram. Now right now, this is a visual comparison game. It's interesting to see what's out there, but it's not exactly rigorous or programmatically accessible easily to you via the web website. The new Reactome platform software allows interspecies comparisons directly in the browser. As for curation, curation is an ongoing process. We have a very dedicated curator, Vindhya Amar Singh, who's working very hard on both rice and Arabidopsis curation. We've got over 200 curated rice pathways that were originally converted from rice cyc data, and then the manual curation and update process went from there. Recently, we put out a release with 16 new ones a couple of weeks ago, 16 new pathways, including auxin signaling and steroid signaling, to give some examples of how we're really focusing on the regulatory and the signaling area of our data right now. On the Arabidopsis side, we converted AeroPsych 11 data and merged that into the general curation database that we share with the Human Reactome project. And the Arabidopsis curation began back in March. To date, we've got about 70 curated Arabidopsis pathways in the central database, and our curators are working on more all the time. I want to emphasize what curation can be a fuzzy word. What does curation mean in our context? A lot of what we're doing is making sure that our pathway data reflects current literature as recent as possible. That's obviously an ongoing game. For example, xylem biosynthesis. But this is a view out of our Reactome Curator tool of an Arabidopsis pathway being actively curated. And on the left-hand side in the upper orange box, you'll see a list of AGI identifiers for Arabidopsis and common gene identifiers for those. And this is an associated paper that our curator found. This was just published in August of this last year, meaning that probably many databases may not have caught up to that published information. She grabbed the content of that paper and made sure that the biology that they're reporting on the basis of experimental evidence is reflected in this examples of other pathways we're working on in Arabidopsis. Now, the new Reactome platform, I'm very excited about this. The Human Reactome project released several months ago their version of the new platform called Reactome 3. Then they followed that up with a rewritten analysis tool suite that was released late this last summer, about August. And we've been in the process of configuring and making their software work with our data well so that we can release that. I have this running in a local development environment that is behind firewall, so I will not be demonstrating today, but I took screenshots before I left. We have it running in our dev environment with our released data. So I have all of our projected data, everything we've done in rice as a reference to date, and I'm going to be showing you what this is looking like when we move this to our test in our live environments in the coming weeks. You'll see here our species drop-down list in the new Reactome 3 platform with all of our projected species, including all of those arises. The familiar navigation on the left-hand side, the pathways within a species. Things look a little bit different. This environment has been updated. This is all rendered in HTML5 now, and via APIs we have good programmatic access to this. It makes the release of the data much faster. It also makes it much more flexible what we can do with these diagrams on the fly, and I'm looking forward to building new visual analysis tools with that new software platform. There are also additional tabs at the bottom, some of which I'll show you now. The Atlas baseline expression data, Robert and I at EBI and I have been working together to make sure that our data is coordinating with theirs. This is an example of rice baseline tissue expression data in the root associated with a particular pathway, and the RAP identifiers for the rice genes that appear in this pathway with the baseline expression information. When you pull up the pathway, this just shows up when you hit the expression tab, which is nice. And then there are cross links out of here to the EBI Atlas project. There are APIs, again, similar to some of the things that Paul showed earlier, for the Reactome platform as well. I'm just showing XML examples, but it's also available in JSON. And actually, the entire new Reactome platform runs off of its own API internally. So when these diagrams are being generated, it's going against the same API that's also publicly available to get that data. The analysis tools is a very exciting new area. I look forward to getting out on the test and live site. You can upload your own analysis data. That could be many, many different things. It could be just a list of uniproduct sessions or gene names, small molecules. It could also be microarray data and metabolomics data. So if you've done expression analysis studies, you could upload tab delimited data in this box here or by file, hit analyze, and then you get the results of that back laid directly onto the pathway with expression analysis. I ran this with a test of some diurnal expression data we had in-house. It has time course associated with it, obviously, and there's a little orange box here with some arrows on the left and right that allow you to move forward and backward through the time course to see the progression of expression over time with additional numerical data at the bottom. You can also do that with metabolic data. This is just using, this is not actual experimental data, this is test data that Reactome provides with their analysis tools. I just wanted to show how you get some more variant coloring of expression across the bar for different small molecules, ATP, ADP, water, etc. Also, importantly, we have interspecies comparisons, so you don't have to do visual comparisons by hopping back and forth between pathway diagrams. You can actually overlay one on top of the other. In this case, I overlaid ZMAs onto our curated arousal sativa pathways. The blue boxes mean that this particular gene product was not present in maize. So it's absent into the species we have projected to, but it's present in arousal sativa. The yellow boxes, or partially yellow boxes, show where you have some orthological inference from the first species to the second, and how much is determined on the basis of whether or not you are moving from a single gene product to a complex or a collection of gene products. So the progress of the bar across the box just tells you how many gene products you are projecting to, and whether or not you had the full set or just a percentage of them. If they're a white box, it just means that there's no orthological inference data between the two species as opposed to being distinctly absent. It may just be missing data. I just want to mention in closing that I'm giving a webinar on Plant Reactome coming up on Tuesday, January 27th at 2 Eastern Time, and that we have an entire series of webinars associated with the Gramene project on these dates, and this information will be available, including links to the webinar, on gramene.org. With that, I would like to say thank you to the many people involved with this project. Special attention to Vindhya Amarasinghe and Sushma Naithani for their involvement with curation and outreach, and to Kapil and Andrew over at Cold Spring Harbor working with me to make sure that our Plant Reactome data is indexed on the main Gramene site and active and available. Peter is one of our curational mentors at NYU. I had a lot of help getting the new Reactome platform configured and working with the good folks at OICR and EBI, and some other folks that have been involved with the Reactome project in the past couple of years that have moved on to other positions. And with that, I'd like to say thank you.